Very much, Stephen, for the kind introduction. Good morning, everybody. I would like to thank Alison and the whole organizing committee to give me the opportunity to talk. I'm relatively new to the transfusion community, so we're just starting work using actually products from the blood transfusion service, and I would like to give you kind of my perspective as a platelet researcher on platelet concentrates. And as you can see in the background, we are using a lot of microscopy, and we're not only looking at platelets, we are also interested in general in what is needed to form a stable clot to actually stem bleeding. That's the wrong one. So I have nothing to disclose. And as a short overview, I would like to give you a glimpse of a recent research project which might be of interest kind of in the terms of how we approached the different effects of different blood components for blood material interactions, and then really focus on the questions, what research can we do with and on platelet concentrates. So in our lab, we mainly use blood from healthy donors. So we are interested in studying the platelets either in whole blood, then probably mainly in transfusion, in perfusion assays, in PRP sometimes, but mainly really we are using washed platelets because we would like to kind of circumvent the effects of plasma proteins and anticoagulants on platelet function. But of course, that might mean that we will be missing some interactions between blood components and platelets when, for example, blood interacts with materials. And blood material interactions are not only important when you handle or store blood products, but they are also important when it comes to implants, because every implant first gets in contact with blood, and this contact then later actually determines the response of a lot of other cells actually to the implant. So we have looked at scaffolds which were provided by a collaborator, Dorothea Brüggemann in Bremen, and she produces scaffolds made out of fibrinogen, which she can tune to adopt different topographies, either flat sheets or actually sheets which have a topography very similar to fibrin fibers. And we were asking actually how do platelets react to these different topographies? Is there a difference in how these surfaces actually activate platelets and what the effect of other components is? So when you seed washed platelets on these different substrates, they behave very similarly in terms of adhesion, spreading, and also in terms of, for example, p-selectin expression. When you do the same experiment with PRP, you get a drastic different response, especially when you look at the planar fibrinogen sheets. So while the platelets are still able to spread on the nanofibers, they were not able to spread on the sheets anymore. So to understand better what was mediating that effect, we pre-treated the scaffolds with plasma proteins, washed them, and then seeded washed platelets. And as you can see, so don't be confused by all the colors, but that's the wrong one. Don't be confused by all the colors, but essentially you see that the platelets are still spreading and not spreading, very much like the response on PRP, which tells us that not the interaction of plasma proteins with the platelets, but rather with the scaffold before platelets interact with the scaffold mediate that response, which is in accordance with kind of the common paradigm as well. And in further experiments, we could show that it's really the topography which makes these surfaces more activating in terms of the responses of platelets. And I think this approach to tear apart the different components and recombine them might be very interesting in the context of looking at the function of, for example, platelet-rich plasma products for regenerative medicine. So could we have used, or why don't we use actually platelet concentrates for our normal platelet biology research? So the reason is that we know that as soon as the platelets are taken from the donors, that the functionality actually changes over time. It does so really quickly in whole blood, where it's about 2-hour time frame where you can work with it, PRP 4 hours, washed platelets 6 hours. So it's really short time frames where the functionality changes, and all the guidelines in the platelet biology field kind of prevent us using platelet concentrates because we wouldn't be able to publish our research. So but it also tells you already that this is kind of at the core probably of what is an interesting question in terms of how to store platelet concentrates, because we have to store them and we would like to store them actually as long as possible, probably to really facilitate all the logistics around platelet donations and transfusions. So that brings really to the question that many of you might be interested in, is that what additive solutions actually, how they impact and storage temperature, how those impact on the functionality of platelets. Their ability to stem blood loss, their ability to circulate in the transfused patient, and perhaps also in the thrombotic potential that they might pose upon transfusion. And I will mainly ask, how actually could we assess, what do we have to measure in the laboratory which mostly represents their function in vivo? So that is really the core question, and we have thought about that question not in the context of platelet concentrates a lot over the last 6 years, but in terms of our own research. So platelets, unfortunately, do a lot of different things while they are building a thrombus. They adhere, they aggregate, they contract. At the same time, they are secreting different molecules from either dense granules or alpha granules, and they also kind of promote the formation of thrombin at the platelet surface. And there are many different assays being used, and we use all of them also in our lab to actually look at aspects of these functions. So it's really important to notice we are only measuring aspects, we are not measuring everything. And as you can see also that actually one of the aspects is kind of underrepresented. It's kind of a blind spot of the current measurement techniques, and that is the element of contraction of the platelets. And that's what we mainly focused on, and I think we have very promising results that this is not only complementary to the other assays, but might actually comprise some of the other assays and be a more holistic measurement of platelet function. So why is platelet contractility important? So as you can see here on the left, on the left, it is important for the compaction of the clot, and it's important that the thrombus doesn't embolize, so that it's really stable. And it happens independently of thrombin generation, and that is really critically important for primary hemostasis. So we know, and to measure these things, our group and other groups have developed techniques to measure the contractility of platelets more directly than just looking at clot compaction. And typically all these methods kind of impose that you put the platelets on a substrate which is elastic, and when the platelets pull at the substrate, it will deform, and we can look at those deformations under a microscope. And you can do that on the level of single platelets like we did in our lab, or on the level of small aggregates, where here, for example, this post is bent with the platelets kind of forming a clot and pulling at it. And this research over the last years has shown that impaired platelet contractility is causative for bleeding phenotypes where the myosin gene is affected. It's prevalent also in other platelet function defects and in some patients who have bleeding of unknown cause. It is a hallmark of the action of antiplatelet drugs in vitro and in vivo. And it is, very importantly for this audience, indicative of the bleeding risk of trauma patients. So we really think that it is a more holistic measurement of function. It's especially important for the hemostatic potential of platelets. So we don't think it really plays a big role for the thrombotic potential, but that also needs to still be clarified. So we think that there is room for testing biomechanical functions of platelets in platelet concentrates, and this might inform us about their hemostatic potential. So this hasn't been done, so that's what we will be doing. What has been done, what's the next closest thing to that, is that people looked at the passive biomechanical properties of platelets, and that's quite promising. They used a technique which is called real-time deformability cytometry, which is essentially a imaging flow cytometer with a constriction where the cells, when they pass, deform, and you can measure how stiff they actually are. And what they found is when they looked at platelets from mice, either wild type or mice with this mutant in the myosin gene, is that normally they have a certain flexibility, but they become much stiffer if they actually have a defect in their contractile apparatus. And very interestingly is that when the same technique is being used to look at stored platelet concentrates at room temperature or 4 degrees, that you see also at 4 degrees, which of course has the promise to prolong the shelf life of platelets, but you do see that there is a defect here related to the deformability, which is probably very directly related to the cytoskeletal defects that are known to happen upon cold platelet storage. And interestingly enough, this group showed that adding a higher concentration of magnesium might reverse some of these changes. So lastly, the second question is, the problem with cold stored platelet is that when you transfuse them, half of them are probably filtered out already on first pass. So they're not circulating for a prolonged time, and that is a problem and hindering the usage of cold stored platelets. And key hallmark research already 15, 20 years ago by Katrin Hofmeister has shown that causative for the clearance might be the clustering of von Willebrand factor receptors on the platelet surface, which are called GP1B. And this then leads to both the recognition by macrophage receptors and their clearance, as well as by higher binding to von Willebrand factor and shear-mediated clearance. And the next, so this hasn't been looked at. That's also something we want to do now with platelet concentrates. But the next best thing again is that people looked at associated changes in the cytoskeleton. And I just wanted to highlight a recent paper which did so in a high throughput method with imaging flow cytometry. So they looked really only at the shape really of the platelet, which is a very coarse way of approximating these defects. But they could very nicely show that cold stored platelets kind of did lose their normal lentil, I always call it lentil shape, so the discoid shape of the platelets very quickly. So in summary, I think we have to be aware that no matter what we do, a lot of things are changing when we store platelets, no matter how we store them. And the important thing is really to understand which of these properties is really of functional importance and which property we have to maintain when we try to optimize our protocols for platelet storage. So as Richard Feynman, a famous physicist and Nobel laureate and a very funny guy, actually said there's plenty of room at the bottom, in this case, for research on platelet concentrates. And I'm very happy that IBTS actually has agreed to form a collaboration with RCSI, where I work, to really look into cold storage of platelets and actually ABO blood type related questions. And we have 2 postdoc positions. The deadline for these is next week. If anyone here is interested or knows someone who might be interested to join this effort, very welcome. I'd like to thank the people, especially my PhD student Martin Kenny, who did the work on the scaffolds, and you for your attention and your kind invitation.
Thank you very much, Ingmar. The paper's open to questions. Yeah, Andre.
Very nice presentation. I was just thinking, about the platelet origin. Okay, so the megakaryocytes. The megakaryocytes. So the cytoskeleton. Because basically what your biomodel about the changing of the shape. That's what you're looking at. It's all the acting, the convolution, the involution of the megakaryocytes, and how the megakaryocytes create the platelet, and it's the shedding of the platelet. And then, obviously, the platelet generate microparticles. So you're measuring the shape. Did you measure the functionality, which is indirectly, the microparticle generation? That's the question. Did you see any change in microparticle generation in cold versus with a different temperature level?
So first of all, we are just starting the work on different storage conditions. We still have to sign the collaboration agreement before we can actually appoint someone. But secondly, we haven't looked in our research a lot on microparticle generation. We do see it happening, of course, when we see platelets, for example, on collagen substrates. It's very well known that they start to balloon and then form these microparticles and shed them. We haven't analyzed them in detail. I think your first introduction was more related to that megakaryocytes form platelets. We are very interested in that process. it probably is a bit then distinct kind of from what happens with the platelets over longer time, but related, because some people also showed that if you would have platelets at 37 degrees, that sometimes they still kind of divide like they do in the circulation when the pro-platelets form before they're kind of becoming mature platelets. So this is something we had looked into, and probably that might be related then to looking at platelet counts in platelet concentrates. I think there is some indication from a poster that we saw yesterday that platelet counts might even rise in the beginning, which is kind of might be related to this kind of idea of platelets creating progeny, which is very controversial actually in the platelet field.
Any other questions? In fact, Andrea, I was thinking of you when this talk was happening and thinking about the growth factors associated with platelets too. And I know they've been utilized in many different ways. Have you done any work in respect of these growth factors?
So release of growth factors mainly from alpha granules. We have not looked a lot at the effect of growth factors on other cells, if that's what you're thinking about. So another group in RCSI, which is more in the tissue engineering field, Fergal O'Brien's group, is looking a lot into using PRP for regenerative purposes, and that's where we are collaborating. And they have very nice papers where they study, for example, using PRP for bone defect healing and looking at, for example, osteoblastic differentiation, indicative of growth factor release. And they look at the growth factors in more detail. We have submitted a grant proposal, and if it's funded, we will be looking at something quite similar.
Lovely, thank you.
Thank you very much. Fascinating talk. One of the points that to me always surprised me is the anti-inflammatory role that sometimes the lysate platelet plays. And I never had understood or had actually even go through the literature to see. Do you have any explanation or line on that?
For
if you lysate the platelets overly and you put a platelet lysate product.
So you say for anti-inflammatory or inflammatory? So I think that's an open question, especially in the PRP kind of regenerative medicine field, because you have formulations which are leukocyte-rich or poor, and I think it has been shown that the leukocyte-rich formulations sometimes lead to excessive inflammation, actually, so exactly the reverse that you're saying. So you're right that platelets themselves kind of seem to maintain that balance, and they're immune cells from their origin themselves, but I'm not an expert in that, unfortunately.
Thank you.
Like in most of the things, unfortunately.